Hi, hi everybody. So yeah, good morning. Today let's talk on the topic ELISA. Okay, ELISA. Yeah, I'll share the screen. 1 second. It's getting recorded. Yeah. Okay. I hope all of you can hear me. Yes. Hmm? Okay. So today's topic is ELISA. Okay. This is a topic in microbiology. For all the students writing the exam, be it MBBS or paramedicos, ELISA is one of the important questions on your micro paper. Probably a 5 mark question, or maybe a 3 or 2 mark question. But it's a short question. Okay, so this topic is little difficult to understand, but I'll try my best to make it easy. Okay, so there is a little bit of mugging up in it, and it's basically conceptual. Okay, but by the end of the class, if you're not understanding the concept, I'll tell you 4 easy points. Okay, so in case this question comes, just write the full form, 4 easy points, and draw a picture. First, so you will get good marks. I mean, out of 5, 2.5. So if the teacher is really nice, she'll put for you. So let's start. I'll stop the class maybe within 20 minutes. Okay. Yes. So let's start. Okay, so what is ELISA? First thing, you should know the full form. What is ELISA? It is enzyme linked immunosorbent assay. Okay, so it is enzyme linked immunosorbent assay. It is also called as EIA. What is EIA, guys? Hmm? It is enzyme immunoassay. Okay. So in the question paper, if EIA comes, write the note for ELISA only. Okay, don't get confused. Fine. Now, tell me what is this ELISA? Yes, you're right. It is a diagnostic test. Just like how we do a blood test to understand if the patient has a disease or not, let's say TB or HIV. It's a diagnostic test. ELISA is a diagnostic test to find out diseases like HIV, TB, or rotavirus, hepatitis. A lot of diseases we can find out by doing ELISA. Okay, so ELISA is done on a plate. Okay, I'll show you the picture by the end of the class. Okay, it is basically done in such a plate, okay, where this plate has got so many small depressions like this. Tell me, this plate has got lot of small depressions like this, which we call it as well. Okay, you can read. Okay, this we call it as well. Okay, so in one ELISA plate, we have got about 96 wells, mostly on the ELISA plates we have 96 wells. Okay, whatever question. Fine. Now, okay. The second point that you should remember is that all these wells are coated with a material called as immunosorbent material. Okay, immunosorbent material. Now, what is this material? You know, this ELISA test. Now, what are we detecting in ELISA test? We are detecting if there is an antigen or an antibody of a specific disease in the patient. Okay, that is what we are finding out in ELISA. So to see these interactions, we need a material on the well that will stick the antigen antibody to it. Only then we can see the reactions happening now. So that material, which acts like a glue and sticks on the antigen antibody, that material is called as immunosorbent material. This much clear? Okay. Now, before I tell you, so basically in ELISA, na, so what you do is, yeah, you just write the full form and you write what is it? Okay, that is a diagnostic test you write. Then you draw the picture of this plate, write 96 wells are there. And then, now to understand the four basic steps, we have steps of doing the ELISA. You should know certain terms, okay? What? Because if you don't know these terms, when I tell you the steps, then you won't understand. Okay, so basic terms first you try to understand. Hmm? Okay, so what is antigen, guys? Tell me. What is antigen? Antigen is anything antigen is a protein that causes infection. Okay, in simple terms, if I tell you, it is that. So let us say I'm taking a Mycobacterium tuberculosis bacteria. Okay, Mycobacterium I'm taking. Now this Mycobacterium, and I'm just giving you an example. Okay, so it will have a protein, okay, which I call as antigen. Antigen is written as Ag. Okay, which I call as antigen, and this as such, the bacteria is not the problem. The protein in this bacteria, that is the antigen in the bacteria, when it enters a human body, this antigen is causing all the problems in the body. Understood? Okay, so the antigen is a villain. Okay, so this happens for any virus also, any parasite, all these will have this antigen, and this antigen, when it enters our body, this antigen is a problematic stuff. Okay, so if we kill the antigen, then that organism becomes non-infectious. Understood? So that is the meaning of antigen. Antigen is present where? In the microorganism. Okay. What is antibody, guys? What is antibody? When antigen enters our body, our immune system is a fighter. Okay, so this immune system produces fighters against the antigen called as antibodies. Ye to all of you will know. Okay, now what is this enzyme linked antibody? See guys, in our labs, okay, in the micro labs, we can create antigen antibodies. Okay, so I'll just give you an example of HIV today. Let's take example of HIV virus. Okay, in the lab we can create antibodies. Antibodies I write as AB. Okay, antibodies against HIV. Okay, we can create antigens also that is similar to HIV. Okay, so we have manufacturers, people who create synthetic antigens and antibodies of certain diseases. Okay, because we need these to perform certain reactions to find out if the patient has disease or not. We need these synthetic antigens and antibodies to for our ELISA test and other tests we have. We need these synthetic antigen and antibodies. Okay, in the lab we need them, so manufacturers produce it and give it to us. Okay, now we have these normal antibodies against diseases. Those are also produced by the manufacturers, and we have something called as, I'm drawing like this, an antibody. Okay, so if this is an antibody, these manufacturers will stick a enzyme near to the antibody and give us. That also we're using for certain steps. So such an antibody is called as enzyme linked antibody. This is a basic understood. If you didn't understand, it's okay. When I tell you the steps, it'll be clear for you. So yeah, just hear me out once, and when I say the steps, you will understand it much better. Okay, now tell me one more thing. What is substrate? What is substrate, guys? When we do certain reactions, okay, like in the test tube or in a slide, one second. Yeah, guys, when we do certain reactions, some reactions we can't see through our naked eyes. Okay, we need something to tell us ki the reaction has happened. Okay, so for that we use substrate. Okay, so I'll just give you an example. Suppose I put an antigen antibody in a test tube, but with my naked eye, I can't understand if they're bonded or not. Okay, if the reaction has happened or not. So what I do is, then if I add a substrate, and then if I add an enzyme, okay, this enzyme will break down the substrate, and whenever substrate is broken down, it produces color. Okay, this concept you understand. If I put a substrate in a test tube, okay, in a normal test tube, and if I put a enzyme, okay, this enzyme, each substrate has its own specific enzyme. So if we put those two together, substrate and an enzyme, this enzyme will break down the substrate, and whenever substrate is broken down, it produces colored products. This concept understood. The rest also forget. What is substrate, guys? If it is broken down by an enzyme, it produces color. Okay, this concept understood. Oh, very good. Now we'll move on with ELISA. Okay, so ELISA test, now we have 3 types of ELISA. So ELISA test is for what, guys? If somebody asks you what is ELISA test, what are we detecting in ELISA test? In ELISA test, we are finding out if there is antigen or let us say antibody against if there is antigen or antibody of a particular disease in the patient's body. That is what we are detecting in ELISA. Understood? Okay. Now we have different types of ELISA. Okay. Depending on our need. Suppose if we want to find antigen, we can do a specific test. If we want to find antibody, we can do another test. So basically, we have 3 types of ELISA. Okay. First, just learn the name. We have 3 types of ELISA. One is sandwich ELISA. The other one is indirect ELISA, and next is competitive ELISA. Just hear the name once. Okay. Now, sandwich ELISA is used to detect antigen. Okay, be it any disease. If I want to know that disease ka antigen is present in the body of the patient, then I use sandwich ELISA. Now, if I want to find antibody, if in a patient's body, if I want to find if an antibody against a disease is present, like if I want to know if the patient has antibody against HIV disease, then I can do either indirect ELISA or competitive ELISA. Both are used to find antibodies against a specific disease. I can use any of these, indirect or competitive. This much clear, guys? So this much is clear. Now, you have to study all these three. You should know how to do the test. You should know concept, and at last you should know the inference. Okay, so yeah, we'll start with the first one. It is simple. Okay, it's not like when I studied my MBBS. To tell you the truth, I didn't understand ELISA. I don't think when I don't remember studying this thing for my exam, but it's a short question. So yeah, once I understood the topic, now I thought that I should not let it go without teaching you all, because the concept is so clear. It's easy. Okay, so let's start, guys. Before I tell you how to do these three types of ELISA, you should know these. 1 second, guys. Okay, so yeah. So before I tell you the 4 steps of doing ELISA, you should know. No, sorry. Uh, First, yeah, you should know these 4 rules of ELISA. Okay, these are the 4 basic rules of ELISA, the steps of ELISA. The first thing is, I told you that in a slide. Okay, we are doing it, not on a slide, we are doing it on a plate. Okay, and this plate has got so many wells on it. This also I told, wells matlab depressions. Okay, now if I take so for illustration purpose, I'll take one well and draw it like this for you to understand. Okay, so basically, why we have 96 wells, guys? Because we can check many samples simultaneously. Okay, each if I check a sample, a patient A ka sample in this well, I can put the patient B ka sample in this well, C ka sample in this well. So simultaneously, I can check many people's result. Understood? Okay. So the first thing is, if you want to find antigen, if you want to find if an antigen is present in the patient's body, let us say HIV antigen, then you have to coat the well with antibody. Understood? First step. If you want to check if an antigen is present on the patient's body, then you have to coat the well with that specific antibody. Matlab, if you want to see if the patient has HIV antigen, then coat the well with HIV antibody that you have in lab. This much clear? Now, vice versa. If you want to check for antigen, I'm drawing antigen like this. If you want to check if an antigen is present on the patient's body, then you have to coat the well. Then you have to coat the well with opposite. What? You have to coat the well with antibody. Understood? This much understood? Okay. Now the second step is onto the well. Okay, onto the well, you add the patient's sample. The second step is, once you coat the well with antigen or antibody, you add the patient's sample. Understood? Okay. Third is what? You add the enzyme linked antibody. Okay, so in the lab we have enzyme linked antibodies already prepared. Okay, suppose I'm checking for an HIV patient. I have preformed HIV antibodies, but these are linked with an enzyme. Okay, so the third step is you add an enzyme linked antibody. Clear? Okay, and the fourth step is you add a substrate, and the fourth step is what? You add a substrate. So these 4 points you should learn it right now. Okay, at the end of the class, if you don't understand anything in ELISA, in your answer sheet you write these 4 basic rules. Okay, and draw the plate bus. Okay, so you see, these 4 rules are very simple now. First, coat the well with antigen or antibody depending on what you're finding. Okay, if you want to check for an antigen, you coat the well with antibody and vice versa. Second, add the patient sample onto the well. Third, can you hear me, guys? I'm having a little bit of fever and sore throat and conjunctivitis. So yeah, the system is a little down. Okay. So second is what? Add the patient's sample. And the third one is what? Add enzyme linked antibody. And fourth one is add the substrate. This much clear, guys? Okay, if you understand this, we can go quickly on to the steps of ELISA. Okay. So first one is what, guys? Sandwich ELISA. First one is what? Sandwich ELISA. In sandwich ELISA, what are we finding? You will tell me what are we finding. Are we checking for an antigen or an antibody? I told you in sandwich ELISA, we are checking for the antigen of a particular disease in a patient. Here, for example, okay, in all three types of ELISA, I'll be giving examples of HIV patient, okay? Suppose you want to check if a HIV antigen is present in a patient, okay? Then how will you do? Then what will you do out of the three ELISAs? Which ELISA will you choose? If you want to find the antigen, you will select the sandwich ELISA. Okay. Now just follow these four basic rules. It's gonna be a cakewalk for you. Okay. So yeah, the first step is what? You are going to find antigen. What will you coat the antibody as? What will you coat the well with? What will you coat the well with? Yeah, you will coat the well with antibodies of HIV. First step. Clear? Okay. Second is what? Yes, second is what? Add the patient's sample. So I am adding the patient's sample. Okay. Now tell me, guys, if the patient is actually HIV positive, okay, this patient, poor guy, has HIV, okay, his sample, okay, you can usually, for ELISA, what sample do you take, guys? You take serum. You take serum, or you can take CSF also, but mostly serum is what you take, okay, blood sample. So you add the patient sample. Okay. Now, if the patient is HIV positive, in his serum, there is going to be HIV antibodies, right? Okay. I'm sorry. If the patient is HIV positive, here we are checking for antigen. If the patient is HIV positive, in his serum, there is going to be HIV antigen, right? Okay. So there is going to be definitely in his serum, there's going to be HIV antigen and HIV antibody. Yeah, in this serum, there is going to be HIV antigen for sure. You know what this antigen does? This HIV antigen will come, okay, and bind with the antibody. Clear? Because, antigen antibodies are like lovers, okay? So if you place a HIV antigen and HIV antibody together, they will bond. They will form a strong bond, okay? So yeah, so the antigen in the patient's sample will come and bind with the antibody. Will bind with the antibody. Clear? Okay. Now listen, guys, I forgot to tell you one step. After each step, okay, out of these four steps, after each step now, we do washing. We wash the plate. We wash the wells. Okay, so we wash the plate so that, if suppose there is no bond being formed, okay, any extra antigen or extra antibody, we just need to wash it off. That is why. So just remember that after each step, we wash the plate. This point you should remember. Okay. So I added the patient sample and I told you if he is positive, his serum is going to have what, or CSF is going to have what? Antigen, HIV antigen. This antigen will form a bond with the HIV antibody that you have coated on the well. Clear? Very good. Third step is what? Add enzyme linked antibody. Okay, so now I am adding an enzyme linked antibody here. Okay, I am adding an enzyme linked antibody here. This is the enzyme linked antibody. The black dot that I have drawn is the enzyme. So in my lab, I have synthesized what? Enzyme linked antibody. So I am adding on to the well. This much clear? Okay. Now, see, I told you this antibody, okay, also likes what? This antibody also likes our HIV antigen. I told you whenever you place antigen and antibody together, they'll form bonds. So this antibody will also form the bond with this antigen. Clear? Okay. Now see, guys, it's like a sandwich. Okay. On top, you have one antibody. In the middle, there is one antigen. Below, there is another antibody. So it is formed like a sandwich where, there are two slices of bread on the top and the bottom. And middle, there is a chicken patty. Okay. I'm a big fan of sandwiches. What about you guys? Okay. See, guys. So this forms like a sandwich. Okay. And that is why we call this ELISA as sandwich ELISA. This much clear? Okay. The fourth step is what? Add a substrate. Add a substrate. So I'm adding a substrate. Where is, wait a minute. Okay, I'm just adding a substrate here. Let us say this is a substrate S. Okay. So what does this enzyme do? Okay, this enzyme will come and break the substrate. So when the substrate is broken down, I told you what will be formed? Color will be formed. Okay, so any color depending on what enzyme and depending on what substrate you use, the color can be formed. Sometimes could be red, green, depending on what enzyme and substrate you put. Okay, but definitely you can see a color change from transparent, okay, or from slight yellow color. You can see a color change. You can see red, green, depending on the enzyme and the substrate you have added. So color change is there. So whenever you see a color change, it means that the patient is positive. This much clear, guys? This much clear? Okay. Okay, now tell me, suppose guys, uh, you have taken, this is a well, okay, and you're suspicious that if a patient B, okay, if patient B has HIV or not, okay, and you want to check for HIV antigen. Doctor has written to check if he has HIV antigen or not. Okay, so since it is antigen, you want to use a sandwich method. Okay, now I'm telling you, the doctor has suspicion that if the patient has HIV, but I'm telling you ki now the patient who's come to you is normal. So if a normal person comes to you and you do sandwich ELISA, what is going to be a result? I'll tell you. So first, what you're going to check antigen. What will you quote the? Now you will tell me the answer. Okay. What will you quote the well with? Very good, guys. You will quote the well with antibody, right? HIV antibody. Second step is what? Add the patient's sample. Now, if he is normal, will he have HIV antigens? No, he won't have HIV antigens. So will there be anything to bind with this antibody? No. Okay. Now, third step is what you're adding? Enzyme-linked antibody. So I'm drawing an enzyme-linked antibody. Okay. Now see, will these, what happens is, now tell me, will these two antibodies form a bond? No, antibodies and antibodies won't usually form a bond. Okay, they are like repels. So once I told you after every step you're washing, okay. So after adding enzyme-linked antibody, definitely there is no bond being formed. Okay, 1 second, guys. There is no bond being formed, and when you wash now, just a moment, when you wash, this enzyme linked antibody will be washed away. Okay, and the last step is what? You add a substrate. Now to break down the substrate, do you have enzyme here? Because enzyme linked antibody got washed away. You don't have you don't have an enzyme. So will the substrate be broken down? No. Will there be color? Obviously there's not going to be any color formation. Okay, so this is a normal person. So if you didn't understand this, it's okay. Just remember, in sandwich ELISA, if there is a color change on your well, it means the patient is positive for that specific disease you're checking. And if there is no color change, it means that he's a normal person. Easy? Okay. Next, guys, yeah, just 5 more minutes to go. Fine. Let's do the second kind of ELISA. What is it? Second ELISA is what, guys? You tell me. Very good. Indirect ELISA. Okay. It is indirect ELISA. I think my baby is crying. Just a moment, guys. 1 second, guys. 1 second. Indirect ELISA. Okay. So yeah, the final two topics, indirect ELISA and competitive ELISA, will finish it off hardly like 5 to 7 minutes. So what is indirect ELISA, guys? What are we detecting? Now you tell me the answers. You know the basics. You will tell me the answer. Okay. Fine. So what is indirect ELISA? What are we detecting? Trying to detect? We are trying to detect the antibody of a patient. Okay. So here I'm taking example of HIV patient. Okay. HIV patient. Fine. So I'm drawing the well here. Okay. And let us say if okay, the doctor tells you that he is giving you a patient A, okay, and the doctor is telling you ki dekho, I think the patient is positive for HIV, check his antibodies. Okay, so what you do? Okay, you will go and do indirect ELISA, or you can also do competitive ELISA, because both are used to check antibodies of diseases. Clear? So first, how will you do indirect ELISA? You can do both actually for double confirmation of the disease. Okay, so how will you do indirect? First one is what? First, the concept is here you're trying to find the antibody of HIV. Here example is HIV. Okay, so the first is what? First, what will you coat the well with? Now you will tell me the answer. Okay, you will coat the well with Very good. Opposite. Okay. Antigen of HIV. This is the antigen. This dot I've drawn here. It is the antigen of HIV. Okay. What is the second? So first step is done. What is the second step? Add the patient's sample. Okay. So here I am adding the patient's sample. So, if the patient is actually positive for HIV, actually, if the patient has HIV, his sample will be having what? HIV antibodies now, right? His sample will be having HIV antibodies. So, I am drawing. So, this HIV antibodies now will go, okay, and bind with what? This HIV antigen. Okay, because antigen and antibodies are lovers. This much clear? Okay. Now, so the second step is also done. What is the third step? Okay. So remember, after every step, you're washing the well. Okay. So tell me, after adding the patient sample and I wash the well, will this antibody get washed off? No, because it has formed a strong bond with the antigen. Huh? Okay. The third step is what? You add the enzyme linked antibody. Okay. I am drawing the, okay, I am adding the enzyme linked antibody here. Okay. So this enzyme linked antibody I've prepared from the lab, okay, for the HIV, and I'm adding it to the well. Okay, now remember this line, what I'm going to say very clearly. In indirect ELISA, the enzyme linked antibody that I'm adding is made in such a way that it gets attracted towards another antibody. Again, in indirect ELISA, the enzyme linked antibody I'm adding from the lab to the well is made in such a way that it gets attracted towards another antibody. Usually antigen and antibodies are lovers. Now they get antigen and antibodies, they form the bonds. But here indirect, remember the concept is the antibody forms the bond with what, guys? The antibody, the enzyme linked antibody forms bond with the another antibody. Okay, so this concept you understood. So now see, all these three are in layers. Okay, the enzyme linked antibody, yeah, the antibody of the patient, okay, and the antigen. So all have formed a bond. Okay, now if I wash in this step, will this enzyme linked antibody get washed away? Will it? No, because it has already formed a bond, right? So it won't get washed away. It is there in the well itself. And what is the fourth step? Add a substrate. Okay, I am adding a substrate. Okay, let me take a different color for that. Okay, so I am adding a substrate. So what will this enzyme do, guys? This enzyme, it will go and break the substrate. You all can see, okay, it will go and break the substrate, and once the substrate is broken, it will produce a product which is colored. Okay, it will produce a colorful product. So in short, if you see a color, it could be any color depending on what substrate and what enzyme you're adding. It can produce any color. So if you're seeing a color change in your well, okay, it means that the patient is positive for that specific disease. Clear? Clear? Okay. So, okay, now tell me what happens if, okay, so another scenario, okay. This was patient A. Now I'm taking up patient B. Okay. So let me draw the well again. So the doctor tells you, देखो, I think the patient B has HIV. You just check if he has HIV antibodies. So again, I'm doing an indirect ELISA test. Okay. Now I'm telling you, actually this patient is normal. Okay. The doctor is suspicious, right? But I'm telling you that the patient is normal, but the doctor is suspicious. So he has asked you to check the antibodies of HIV in the patient. Okay. So now how will this step go? Okay, I'm checking the antibody. So what will I coat my well with, guys? You tell me. Very good. I'm coating my well with antigen of HIV. Okay, fine. Now what is the second step? So this step is done. Fine. Now what is the second step, guys? Yeah, add the patient's sample. So I'm adding the patient sample. If he's a normal patient, will he have antibodies of HIV? No, he won't have. So will there be any antibodies that will come and bind to this antigen? No, right? So there'll be no antibodies. Okay. Now what is the third step? Add enzyme linked antibody. Okay, so I'm adding enzyme linked antibody here. Okay, that is my third step. Now I told you this antibody, okay, in indirect ELISA, the enzyme linked antibody I'm adding to the well is prepared in such a way that it is attracted only towards another antibody and not to an antigen. So now you tell me, will it form a bond? Tell me, will it form a bond with this antigen? No, because it doesn't like antigen. It forms a bond only with another antibody. So there'll be no bond formation. This enzyme linked antibody is free to roam, right? Now see, after this step, after every step I'm washing now. So after third step also what? I'm washing the well. So when I'm washing the well, this free enzyme linked antibody will get washed away. Clear? Okay. Now the fourth step is what? I am adding a substrate. Okay. I am adding a substrate. Now to break down the substrate, do I have an enzyme here? Do I? No, I don't have an enzyme. So will the substrate be broken down? No, substrate will not be broken down. Will there be color formation if substrate is not broken down? No, there is no color formation. So basically, if you see no color change in your well after you do the test, it means that the patient is normal. So he can be happy. This much clear about indirect ELISA. Okay, fine. So indirect ELISA is also done. So basically just remember in indirect ELISA, if you didn't understand what I said, if there is color change, the patient is positive for that particular disease, whatever you're checking for. And if there is no color change, okay, if it still remains transparent or that serum color is only there, then it means the patient is normal. Okay. Okay. Now the third one is what? Which type of ELISA, guys? You tell me. What is the third type of ELISA? Very good. The third type of ELISA is called as competitive ELISA. यहाँ तो competition होने वाला है. You know, competitive ELISA. Fine. So what is this competitive ELISA? I'll tell you. So let me draw the well again. Let me draw the well. Here also in competitive ELISA, what are we checking? Tell me, I told you in the first slide, in competitive ELISA, we are checking for the antibodies of a particular disease. Right, okay. So, okay, here also we'll take the example of HIV only. Let's say a doctor tells you that he's sending you a patient A, okay, and the doctor thinks that he's positive for HIV. You check for HIV antibodies in his serum and let me know. Okay, so you tell the doctor, okay doctor, I'll check and tell you. First step, you coat the well with what? You're checking for antibody, you coat the well with, very good, you coat the well with HIV antigen. Very good. Okay, now what is the first step is done. Second step is what? Add the patient sample. Okay, so I'm adding the patient sample. Now see, if actually he is positive, let's assume that he is positive. Patient A is a HIV patient. If he is positive, will he have antibodies of HIV? He will have, na? Okay, very good. So this antibody will come and form a bond with what? The antigen. Okay, the patient's antibody is normal. It is attracted towards antigen only. Okay, normally, universally, antigen and antibodies are attracted. Okay, so remember that, in competitive ELISA, everything is normal. So if the patient is actually having HIV and you're adding the patient serum, his HIV antibodies will come and form a bond with this antigen. Clear? Strong bond. Okay, fine. Now we are washing again. After every step, you're washing. Now if you wash, will this antibody go away from the plate? No. It will be still bonded with the antigen, the strong bond here. Okay. Okay. What is the third step? You are adding enzyme linked antibody. Okay. Let me add an enzyme linked antibody. Okay. Now in competitive ELISA, we are adding enzyme linked antibody. This antibodies is like a normal antibody. It likes to form a bond with what? It likes to form the bond with the antigen only. Okay, it's like a normal antibody. This enzyme linked antibody wants to be friends with our antigen only. It does not want to form the bond with this antibody. No. Okay, but in indirect ELISA, the enzyme linked antibody we added wanted to form bond with another antibody. Okay, that was the principle there. In competitive ELISA, just remember that it is normal and the enzyme linked antibody wants to form bond with our antigen. Okay, but you know what happens here? Okay, the HIV, this one, okay, this one, 1 second guys, the patient ka antibody is so strong, okay, and it will tell the enzyme linked antibody that I am not giving the antigen to anybody, I want antigen all to myself. Okay, so this bond here is so strong that this patient ka HIV antibodies will not allow our enzyme linked antibody to form the bond with antigen. Okay, so it will remain free. This enzyme linked antibody will remain free in the well. Now when we wash the well, okay, so after the third step also we are washing the well. When we wash the well, this enzyme linked antibody, since it is free, will get washed away. Okay, now fourth step is what? I am adding a substrate. When I'm adding a substrate, see, will the substrate be broken down? Because no, because the enzyme linked antibody got washed away. Now there is no enzyme in this well to break down my substrate now. So substrate will not be washed. Substrate will not be broken down. So will there be color production? No, there is no color production. Very important point. So see, if the patient is positive, listen carefully. If the patient is positive, then there is no color change in competitive ELISA. If you didn't understand the concept, it is okay. Just remember this thing. In competitive ELISA, if there is no color change in your well, it means that the patient is positive for a particular disease, for the disease that you have checked. Okay? Now look, suppose, let us say, okay, so this is patient A. Now let us say, let us assume, okay, let's take a patient B, okay, patient B, okay. Now the doctor tells you that, I think the patient is having HIV, just check his HIV antibodies if it is present or not. Now I'm telling you this patient B is normal, okay, he's normal. Fine, so let us see how the steps will go. Suppose he's normal. First, I'm checking for the antibodies. Now I'm doing competitive ELISA. Doctors asked me to check the HIV antibodies in the patient. So I'm going for a competitive ELISA. Okay, so I'm coating my well with what? Tell me. I'm coating my well with antigen. Very good, HIV antigen. Second step is what? Add the patient sample. I'm adding the patient sample. In the patient sample, will there be antibodies, HIV antibodies, if he is normal? No. So there are no HIV antibodies to form a bond. Okay. So second step is also done. What is the third step? Add enzyme linked antibody. I am adding enzyme linked antibody. Now I told you this enzyme linked antibody, yeah, it's like a normal antibody. It likes to form bond with what? Our antigen. So it forms a bond with the antigen. Okay. So now they are like, very thick friends. Okay. Now when I wash, will this enzyme linked antibody get washed away? Will it? No, because it has already formed a bond. Okay. So fine. Now when I add a substrate, okay, now when I'm adding a substrate, this enzyme here, guys, will go and break down the substrate and will form a color. Okay, so if he is normal, then there is color production. Okay, in competitive ELISA, if the patient is normal, then there is color production. Okay, guys, so just let me brief it out for you. Only the concept you need to know. In sandwich ELISA, okay, in sandwich ELISA, one second, guys. One second. One second, let me just, okay, look here. In sandwich ELISA, if there is color, okay, if there is color being produced, then it is positive. The patient has the disease. Okay. Now in indirect also, same thing. If color is produced, the patient is positive for the disease. In competitive, okay, if there is no color, if there is, one second, yeah. In competitive, if color is produced, just learn it like this, it'll be easy. In competitive, if color is produced, then it is a normal patient. Okay, only this concept you need to, if you didn't understand, just mark this up. Okay, so in competitive alone, if the color is produced, it is a normal patient. Okay, now so that's all, guys. Let's see the picture of one ELISA plate and call it a day. See, this is a plate. Okay, this is a ELISA plate. Dekho, okay, you can see, and you can see a lot of wells, right? And you can see that so many wells are giving colors. Okay, so probably it could be the serum of patients or it could be the color change. Anyways, I just wanted to show you the picture. Okay, yeah, this is the ELISA plate. It has got about 96 wells. Okay, one of your Viva questions. And another question is that what are the 3 types of ELISA? That's also one of your Viva questions, guys. So what are they? Tell me, repeat after me. Very good. Sandwich ELISA, indirect ELISA, and competitive ELISA. Very good. So I think that's all. Yeah, that's all about ELISA, guys, that you need to know. So yeah, let's call it a day. I hope the video got recorded. Okay. Yeah, just let me know if you want me to take any topics. Okay. God bless you all. Bye bye. Take care.